reconnection. So this comes to the problem of what is the neural base of learning and memory. Now you might think that after so much talk about progress in brain research, we should know what is the memory, where is the memory, and what is learning. And I can tell you that we know very little still. Now, where is the memory stored? This is the first thing you would ask, right? Where is the memory stored? The classical question is called, where is the engram? Engram is the memory trace. Where is the engram in your brain? I can tell you, for any memory that we are keeping right now, we don't know where they are. We know that they are probably a synapse, probably a neuron. There's no other structure that's very likely to be in. But we don't know which region this memory, like the memory for you to keep that I'm talking here. This memory, I don't know where it's going to be stored. I don't know which neurons and which synapse are going to store this information. And how is it stored and how is it retrieved? We know very little. We know the activity associated with memory will change the synaptic structure. That LTP is what it is. I'll show you later that you have a structural change, but we don't know where that's happening, right? We know there's change in synaptic strength because we show LTP and LTD. Activity can happen. You can do manipulation in animals, prevent animal to generate LTP. The animal's ability to learn is reduced, right? So you know LTP is important, but you don't know where that LTP is and how it really correlates with the memory, because there are millions and billions of synapses in the brain that could be happening when you have activity. We don't know whether there's changes in connectivity as well as function. Both can happen, but in a real memory, how much is it? Is it changing the existing strength of existing synapse, and how much is the new formation of connection? Right? That we don't know. How does the memory decay with age, with disease? Right? We don't know is the change in the ability to form memory, or is the ability that we can form memory, the memory is not stable. Right? All these are major questions in brain science that we don't know and we need to figure out in terms of understanding the brain and understanding the disease and aging. Right? So I'm going to show you a set of unpublished results that tells you how we address the cellular level problems. How the memory is stored in a specific synapse. So when I said we don't know anything about any memory, now can we find a memory that's really very simple, very specific, and can be induced in the animal reproducibly, and really look for that trace, where that memory is? The best we can find, probably one of the best memory is called fear memory. What you do is you have a rat or mice, you expose it to a sound, pair it with a sound. You can give a conditioning sound stimulus for, let's say, 1 second. At the end of that sound, you give a foot shock. You shock the animal. So you always pair the sound with the shock. So the animal sort of associate these two events together. So then after you do this for a few times only, then you give only the sound without the shock. The animal will freeze, like, as if they're expecting the shock is coming in. Right? So the animal has a fear memory imprinted in the brain. Right? Now the control experiment, you have to pair them together. If you unpair this shock and the sound in a random manner, they don't learn. You give a shock, then you give a sound, and they don't freeze. Right? They have not associated the sound with the shock. So somewhere there's a memory for the sound after this pairing. So you want to look for where that memory is, and what is the change in that associated with that memory. Right? So given the sound that you associate, so first of all, we show that this is highly robust. Right? So once you compare to an unpaired case, the unpaired animal sound produce no freezing. Paired animal, 90% of the time when the sound is on, they freeze. Right? Highly reproducible situation. So where is the memory store for this? We can store for many days this memory. Right? So we want to look for it. Now, the activity of auditory cortex, you might imagine, it might be important because that's where the sound comes in, right? So we do first a simple experiment. We infuse drugs into the auditory cortex, both sides, right? So we block the activity in the auditory cortex, right? Before we apply the conditioning, before we pair the stimulation. Well, the result is that you inhibit the activity by elevating the inhibition, just reduce silence level area. The silence level area blocks the memory. The memory is gone. So activity of the auditory cortex is certainly necessary for producing this fear memory. The another drug we put in is the APV. It's a drug that blocks the plasticity, that blocks the change of the synaptic strength induced by activity, right? So it's LTP basically. If we block the LTP in these two regions, we also wipe out the learning, right? So this was done before the conditioning. Right? You might say, well, if there's no sound activity in the cortex, of course they couldn't learn, right? So the parallel experiment would be to apply these drugs after you have fear conditioning. Right? After the animal has already learned the tricks that produce very high freezing, then you put the drugs in. You silence the cortex. You ask, right after the conditioning, does the activity in the cortex still required? Is the plasticity, LTP, still required? The answer is yes, completely. Right? You still, the activity of the auditory cortex is still required after learning. So somehow activity is still going on in there in order to keep the memory. Right? So this is where we say, well, the auditory cortex somewhere, maybe memory, long-term memory is stay. So now we do some injections. We show that there's a projection from the amygdala. Amygdala is a region where the fear memory is generated. So that actually sends in a projection into the auditory cortex. So, if you inject express fluorescent proteins in the amygdala cells, they sense fibers that goes to the cortex into the auditory cortex. So there's a projection from the center for this fear memory to the cortex. This region of cortex is where we can image with the in vivo the changes in the synaptic structure. Right? So we can image them and compare them with base. The same axon that comes from the amygdala, we can image the axon structure. Now the axon structure has this bouton. Bouton are swellings on the axon that are forming contact with the spines. So it's a presynaptic structure. You can see that there's an increase in bouton. You probably cannot see. The bouton that used to be not there, then that appeared. There are bouton that was there during day one and disappear in later time. So there's a formation of bouton and an elimination of bouton, suggesting there are changes in the synapse formation. Now the trick is to compare the control animal, which has unpaired stimulation where there's no memory, with the paired stimulus, which there's a fear memory. And you'll see that the fear memory, the formation of the bouton is higher. This each point is actually one animal with hundreds of bouton, hundreds of dendrite counted. The average percentage of bouton formation is higher than the control animal unpaired stimulation at 3 days after conditioning. At 2 hours after conditioning, there's already a slight increase, but barely significant. So during this training of pairing, something is happening in this cortex that new connections are made. by the new connection made by forming new butons, new presynaptic structure. The buton that's eliminated or disappearing, constant turnover doesn't change. So it's the new buton that gets loaded in. Now you can, as this specifically happened from that projection, you compare a different projection from prefrontal cortex rather than amygdala. The prefrontal cortex shows their dynamics of buton formation and elimination doesn't change. So that means that one specific pathway related to fear memory formation is specifically increased in a presynaptic structure. We can also look at postsynaptic structure, but this is the spine. The spine on the layer 5 neuron, which sends dendrites up to the surface, we can look at them. This dendrite also increases after 3 days. Now this formation is slow because 2 hours after fear conditioning training, there's no increase in the new spine formation. But 3 days later, there's a clear increase. So there's a progressive increase after training, an increase in the formation of postsynaptic structure. Now you have a presynaptic structure that's increased, postsynaptic spine also increased. You imagine maybe they are together, right? They should form a new synapse of new presynaptic buton contact new spine. That's what we expected. Now, but we don't know because we are separating imaging different structure, right? This is a postsynaptic cells labeled. It's specific to layer 5 neuron, not layer 2-3. Right? But layer 2-3 you don't see changes. So that's again specificity. It's a specific pathway from natural amygdala to layer 5 neuron. That connection, that spine, appear to be increased. So in order to see what's really happening, we need to label both sides. This is the toughest experiment. Now, nobody has been able to do this in the field. We've accomplished to do this. We label presynaptic lateral amygdala neuron with one dye, one color, postsynaptic dendrite with another color. Then we look at their contact, and we ask whether there's change in the presynaptic structure and postsynaptic structure. That's where the new synapses are. So in fact, this is a stable synapse that doesn't change. This is a synapse that was not there, but later on there's a new synapse made by a new buton that was not there before. The new buton making a contact with the spine. Here is again another one. There's no buton here. And then after 3 days, a new postsynaptic structure is formed. So you can look at the newly formed butons, newly eliminated butons, and look at their change. So now, in this case, you can see a clear increase with time. After training, there's a gradual consolidation of memory, we know, during this period, a few days. There's an gradual increase in new synaptic pairs of presynaptic, postsynaptic structure. That's what we call pairs. And the number of existing pair that disappear or eliminated doesn't change. No difference between control and experimental group. That means there's a steady elimination, but that doesn't inherently contain the information of the memory. The information is contained in the new pairs that's formed with time. Now, then we address the interesting question: is the newly appearing buton and newly appearing spine is always that pair? It turns out it's not. It turns out that the new synapses are made by having an existing spine search for a new buton, or existing buton search for a new spine. So you are using the original wire, your original connection. You add new powers to it, rather than you change, you make de novo a new connection. And this is total surprise to us and to many people in the field. Well, this hasn't happened, hasn't published, so I'm sure it's going to be a surprise to the people in the field, because people always thought a new connection is made by presynaptic structure matched with postsynaptic structure. No, that's not an efficient way. If you think about it, there's already a lot of connection there. The way to make a new partner is just add, use the original structure there, add a new partner on it. You save energy, you save resources, you save the space within the system. So rewiring in the adult brain does not involve formation of complete new connection. If it's an existing connection, search for a new partner. So existing synaptic element, search for a new presynaptic partner or postsynaptic partner. That's how the new rules for synaptic rule. And this change in the formation is correlated with the freezing. In individual mice, their level of freezing time is different, highly variable. But if you look at the correlation with the change in the synaptic pair, it's highly correlated. This blue formation is highly correlated. So this supports the idea that the field memory is stored in part in the auditory cortex, and memory storage involves structural rewiring of existing connections. And the interesting thing is that this rewiring is rewiring of synaptic condition in the adult brain to add new synaptic partner to existing synaptic elements. That's how rewiring is happening. This is very different from early development, because in early development you have a large scale rewiring during early postnatal brain.